Okay, I think I'm recording now, so we'll just make a start, and anyone who is joining us a little late has a chance to catch up once we get going on with the chapter material. What I wanted to do today was to first of all look a little bit back on our what we have done in the previous lab and then discuss this week's topic. However, before I get too far carried away, I really wanted to thank you all. You've been working really hard. I saw some excellent lab answers and answers to the exam and so on. So thank you for all that. If you have any questions before we jump onto it, also please feel free to just either type them to the chat or unmute yourself. But if I don't see anything, I'll just move on in just a few moments to share my screen. I see a couple of people are being smart and already doing their attendance. So if you do want to do that, you can absolutely do that at this point. Just post your name to the chat. I'll probably, if we get a chance, I'll also ask a couple of questions to do that as well. But it always helps me that we make a start of that. So if you bear with me, I'll share my screen with you. And you should be at this point able to see my screen regarding this week. So we're at week 5. So let's just have a look through a couple of things before I get too carried away with that, though. Just a quick change that I want to make. I think we should be able to go ahead. Interestingly, I have lost the ability to share a particular app. So what we're going to do, I'm just going to share with you the entire screen and we'll navigate from there. If you do have any questions at this point or later on, because I'm not able to share only one app, I'm sharing the entire screen of my computer. I'm not able to see those individual chat messages. So really the best way is to just unmute yourself and speak up. I'll of course try to take every now and then a chance to pop into the chat and check on that. But just because we're set up at the moment, I'm not able to do that. I just wanted to say a few things about the lab that we completed. There were 2 questions that I think were particularly challenging. So first of all, we talked a little bit about before we get too carried away with the experiment, that sometimes what we end up doing and what we might want to do is to do a baseline. By what I mean by baseline is having some sort of a standard against which we will compare the later steps of the process. And in this lab, we were looking at different substances' abilities to work as a buffer. So what we wanted to see was how that sample and how our analysis of that sample in that beaker filled with distilled water would behave if we didn't have any buffer. So that's really what I mean by when I talk about the baseline and why it was such an important concept for us to have a look at as we got started. And what we saw really with our baseline samples was that there was a significant drop in the pH when we worked with acids and significant increase in the pH when we worked with bases. And of course, there was a limit to that based on what acid and based on what base we used, so that increase was gradual until we reached a point where the changes were very, very small after that. But really just so that we're all on the same page and we're understanding where those points that you got for that lab might have came from. I wanted to clarify that. And on that note, do make sure that you always, when there is a written lab component, you check onto the feedback. I try to get them turned around usually pretty fast, so you're able to see where you either aced it or what else could have been done. So that's just something that I wanted to put out there. The second question that was, I wouldn't say hard, but that caused a little bit of thinking around, was the graphing portion of this lab. And please remember that normally when we do labs on this course, we would spend about 2 hours and 15 minutes together in a class. So when you're thinking of the workload that you're completing for a lab, for example, that's something you could expect it to be. So not all labs are going to be something that you can just race through, and not all labs answers are answers that you will just see while we do our review sessions. Some of them require you to spend a little bit of time playing around, trying your way of figuring out things. And for example, in this case, preparing something that I was expecting to receive. So anyone who tackled this graphing exercise, very, very well done, since it was something that took a little bit of your own time to sit down and figure that out. In terms of the graphing exercise, what I was looking there was really at 2 graphs, one graph for acids, one graph for bases, and I was fairly particular of what sort of elements I'm looking on those. One thing that I tried to work with you whenever I could, but what I do want to highlight, I did ask for them to be sent to me in a picture format. If you couldn't do picture format, you could do PDF or something else, but things that I can open in BlackBoard. So if you sent me an Excel spreadsheet, it might have been that you didn't get the full marks, because if you think about it, that means that I would download everyone's Excel spreadsheets, and I can see the IT challenge with that, but I've been very, very flexible. There were a few students who sent me other file formats, for example, from the MacBooks and so on. If I couldn't get your file open, and I wasn't gonna download every app that has ever existed. If I couldn't get it open, I told you so on your feedback, and you can resubmit that part of the lab to me. But really, if you can, please, 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 always, if I ask very particularly a certain file format or so on, try to do something that works with opening on BlackBoard rather than downloading files. That's part of the learning curve that we're doing here as well. So that's about the labs. Did anyone have any questions about the labs? Are you able to see your feedback from the labs? I won't go on this session to individual people's lab feedback. So if you had some questions about that, I'm happy to stay afterwards. But if you have in general that you don't even know that there is such a thing as individual feedback that you're receiving, I just want to encourage you to make sure that you check on those, because I do take time and care to write you individually feedback. Well, I don't see any questions on the chat. I don't see any hands being raised. I don't hear anyone shouting out. So what I'm gonna do at this point, I'm again gonna move on to another, or to move on back to my screen. And what I want us to do today is to really focus on the lecture material from this week's chapter. And what we're doing, we're looking at cells. So we're going to be talking about that a little bit. So again, let's get going. And you should be able to see my screen. Again, if for some reason you wouldn't see it, chat is not visible for me at this time, so please just unmute yourself and shout out. There's always a chance that things happen. So before we get too far with talking about cells, I think it's important that we remind ourselves of where we are on this course and why we started with all that microscopic stuff before we actually moved on to this chapter's topic. So we are approaching on this course the study of life by looking at different scales, different levels of scales, how we can study life. And we started with the very smallest things. We talked about atoms and then molecules, and we talked a little bit about macromolecules. So these are something that's made of 2 or more atoms when you get a molecule, and macromolecules are then just larger than just your traditional smallest scale of the molecules. So now we get to the cells. So cells are the next level that we will tackle, and cells are really the basic unit of life. They're the smallest thing that we can find that survives on itself, survives without other organelles, other parts contributing to its survival. So when we are talking about cells today, we will zoom into the cell and actually have a look of all the parts that make up a cell. But remember, all of those parts, they individually wouldn't be able to survive. We actually do need to have other parts of the other cell organelles of those to survive individually. So cell is really the smallest level of life that's capable of existing on its own and living on its own. And when we look at the scale of the cells, we really do have a large variety. Most cells tend to be smaller than 1 millimeter in diameter. Some can be much smaller, some can be much larger. I have used an example, I think, on your quiz about cells that play a role in our nervous system and in our spinal cord, those nerve cells. They can be up to 1 meter long as a cell. So they run really all the way from where your spinal cord starts, where your brainstem starts, and all the way down to the lower back. So there is a great amount of variation, but for our purposes, we want to be aware of that on average, cells are small. So that would be the nerve cells that form the spinal cord, kind of an exception. At large, we are looking at small stuff. So when we look at the cells and we talk about their scale, one thing that becomes important for us to also be aware of is how different stuff that we're looking at varies in their sizes. So far, I think that we've mainly focused on talking about the atoms. And we've also had a look of molecules. Well, now we're moving to much bigger things. So we're going to be focusing on cells. And then, of course, at the other end of the scale, if you wish, we could look at things that are a scale of a human, for example. So we are not able to really see cells, or at least the stuff within the cell organelles, with just naked human eye. So to be able to see things that are any smaller than cells, we need to recruit tools that assist us. And those tools that we use are going to be microscopes. And microscopes have a very fascinating, varied history. And I talk about that in other courses of mine with my students. But really what we want to notice is that not all microscopes are light microscopes. So light microscopes are the microscopes that you've typically seen in your science labs, in your schools. These are capable of studying cells and some of the parts within the cell. So we would typically see with a light microscope the nucleus of the cell, which is really dark staining part if you use typical staining methods as H&E and so on. But you wouldn't be able to see much more inside the cell. Well, if I say that we wouldn't be able to see much more, then the obvious question would be that, well, we know that there's much more going on there. So how have we seen that? Well, one approach, and probably the most typical approach for studying things that are smaller than what we're able to see with light microscopes, is by using electron microscopes. And rather than using light beam that we shine either through the sample or to the sample and it bounces back, as we do in light microscopy, electron microscopes use something even smaller. And you guessed it from the name, they use electrons. So those very things that we had a look of at the beginning of this course. So light has properties that causes issues when you go to very, very small stuff. For example, light bends, light can only separate two things that are only so, or that need to be a certain distance apart from each other, but electrons can bypass some of those limits that we have for light. So electron microscopes allow us to study all of the cell organelles, bacteria, viruses, and going to smaller and smaller and smaller scale. I'm very interested on how far we can push this level of scale, and I think that we are getting very far there, and not necessarily on the electron microscopes that we have available everywhere. And most higher ed institutions, until you get to universities, don't unfortunately have an electron microscope available for students to try out, simply because they are so expensive equipment, and it does require a special set of skills to operate those. So we are going to stick with light microscopy, but I want you to be aware of that. You're not going to be one of those students of mine, because whenever I do study cells with my students in a class, they tell me that they're able to see all these cell organelles that we've talked about. And if you're using light microscope, you either are probably having a really good imagination, or you're seeing something else than what you think you're seeing if you're thinking that you're seeing those cell organelles. The other thing that before we dive into the cells that I wanted to kind of talk that relates to what I said, that cells are typically always a certain size. Why are cells so small? Well, there is an actually limit to the size of the cell. And there's few different reasons for that. When you grow a cell much bigger than what it would be typically, what happens is that the ratio between the surface area of the cell and the volume that's inside the cell changes. So what we end up noticing is that we end up with a cell that's no longer really doing its job. It's not able to have enough surface area to meet the needs of the volume that's enclosed inside that cell. So there is a limit to cell size. Another way to look at that, which refers to that gas track that you just saw coming through the screen and stopping there and leaving, that relates to the fact that another way to think of that, what I use with my students is I live, and my campus where I'm based is located on the rural parts of Arizona. And some of the most exciting things that happening here is that a gas truck drives past the town on a highway. Well, if we think that we have a cell, and if the size of the cell grows too large, those organelles still inside the cell are going to need all the stuff that needs to be transported from the cell membrane, from the outside of the cell. If these distances grow too large, those, and I'm using in quotation marks, those vehicles that help us to transport material to those other parts of the cell need to cross such a long distance that they would end up actually burning off or using all those resources that they're there to transport. It's same as there's no point of a gas truck driving a distance that's greater than the amount of gas that it's transporting. Well, at the end, it's burnt all that gas that it was supposed to transport, so no good was accomplished with it. So that's just another way of thinking that my students sometimes find it easier to consider. Well, there are limits to the cell size. However, what we do end up finding that we can push those limits a little bit. And some of the things that we can do if we have a large cell that has surface area that's not meeting the needs of the inside volume of that cell, we can try to come up with creative ways of increasing that cell membrane area. And some of the things that we can do, we can generate various different kinds of folds to that cell membrane folds, or if we're talking of smaller things, these could be microvilli. So microvilli are just much smaller at this level. You can consider it as smaller than what we would have with just regular membrane folds. So on this diagram that I just graphed here, the inside of the cell would be shaded on red. But to make more surface area for the cell, we can make all these kinds of folds to the membrane that encloses the cell. Okay, that takes us quite far on understanding why cells are small and why does it matter. What I would like to do next is that I want to introduce two more terms before we really get to the big topic. The terms that I want to talk with you is that when we are studying cells, we talk about cytology. So in cytology, we're studying individual cells. However, most of the time, if you do go to healthcare carriers, you rarely get a chance to just study one cell. What you end up needing to study a bunch of cells, and those cells typically are very similar in their appearance, or they serve in the same location, or they are part of the same organ, and they form a tissue. So histology looks at the groups of cells that are similar enough that we can call them tissues. So that's really the two main concepts. Did I hear a question coming up, or was that just the microphone picking up noise? Well, I don't hear another question coming up. I had a look of the chat. I'm not seeing anything there, so I'm assuming that we're all still good, and I'm just gonna move ahead with our topics and share my screen with you again. The other thing, just before that brief break and before jumping on to talking about the cells that I promised, that I want to talk is a question that my students sometimes ask me, and this is a very good question. So if we know that we have a cell, and what you're seeing here is just one way of representing cells. This is kind of more of a 3D model or 3D drawing of a cell. And what you can see there is that there's a boundary to the cells. So the cell membrane that we just talked about. Let me go back. So we would see it here. So that forms the outer boundary for the cell, and this cell would be kind of a round in shape. So we're only seeing the sides on this side. There would be sides on at the back and forth as well. Inside the cell, we're seeing nucleus. We're seeing many cell organelles. We're seeing membrane being folded in many ways. We're seeing little sacs carrying stuff. So my students ask me, where did the first cell then come from? And that's a very good question. Okay, if we have this thing that's able to survive on its own only once it has all this stuff inside it, how did it first come about? Because clearly there needs to be a starting point in terms of evolution where it came. Well, what our best understanding at the moment is that the first cell originated as a symbiosis between a cell that didn't have much inside it and then an organism that happened to enter inside that cell just by pure fluke of luck. And there's many theories how that could have happened, but something entered to the cell and it was very successful within that cell. And as a result of that, the evolution worked out. If you wish to think of evolution as an active process, then well, it's handy to have this thing inside the cell. Let's promote this survival strategy. So what I've done here, I've just simply graphed that. So let's say that initially we had only few things or a minimal amount of things inside the cells, but then we found out that, for example, in this example, mitochondria would be useful for the cell to have inside. That would produce energy that would convert the chemicals into ATP that the cell can use to drive many of its processes. But because this was first a fluke of luck, but it was a beneficial fluke of luck for the cell, this became more and more and more prevalent, and eventually we ended up getting all sorts of things inside the cell. The cells are really actually quite busy and quite active places when we look at those. So that really kind of is the introduction to the cells. And what I want to do, I want to remind you that not all cells are the same. There's two types of cells, or most cells we can divide at large into one of the two groups. There's two types of cells that I want you to know. So prokaryotes are these cells that first evolved. They came up much earlier in evolution than the second type of cells that we'll be talking about. Prokaryotes are much more simpler, and they're also, just in terms of their size, much smaller than the second type of cells that we will talk about. What we end up finding is that prokaryotes do have a cell membrane that encloses something inside it. But that's it. There's no other compartments inside prokaryote. In fact, DNA just kind of floats around free inside the prokaryote. Prokaryote does not have a nucleus. So these are the most simpler, the most smallest, the first to evolve. They have very little specialized compartments inside the cell. And these were known as our prokaryotes. The second type of cells that I wanted to speak with you is eukaryotes. And eukaryotes are the cells that you typically tend to think when you think about cells. So these are much larger. There's a lot going inside their specific compartments. We end up finding a nucleus, which is usually this very prominent dark staining structure inside the cell. And that's where the DNA is housed. Like I said, there's compartments, and these compartments contain many other structures that perform many other functions of the cell. So eukaryotes came about later in evolution. And in fact, they are fairly new addition in a bigger scale of the life. And eukaryotic cells are physically much larger than prokaryotes. They have a lot more going inside, many different specific compartments, many different kinds of organelles. And the big thing really that I wanted to highlight here is that the nucleus is enclosed inside the DNA is enclosed inside the nucleus. So one of the things that my students then asked that, well, clearly eukaryotes seem to be like the fancier version. So surely they must be the ones that are winning the race of life, if you wish. They must be more numerous. Turns out that prokaryotes are much more numerous in life. Even though they're simpler and they evolved earlier on, they're very successful in their survival strategy. And we do end up finding a lot of prokaryotes existing and outnumbering the number of eukaryotic cells on our planet. And this leads us to what I've been kind of trying to promise that we will talk about. And we will leads us to talk about these different compartments inside the eukaryotic cells. And that's really one of the main focuses of our class today. Before we jump into that, I want to take a really quick break, and I want to go to the very end of my presentation and just kind of use it as a chance for us to check how much do we remember from what we discussed so far. So I have some quiz questions. I am expecting that we're going to have a lot to go through today. So we're not going to be able to go through every quiz question that might come up in your weekly quiz. Remember that you can take those weekly quizzes up to 3 times, I believe, and you can use your notes, you can use your resources as you take. So it is part of the learning process. It's not just you sending me answers and being done with the course. It is also you taking the time to really learn while you're doing those quizzes. But let's share this screen with you and have a look. How do we feel about some of the topics that we have discussed so far? So far, we have talked about cells being small, and then we discussed that there are smaller stuff than the cell on its own. There's, of course, much larger stuff than the cell. So what I have here is a list of various different kinds of things what I was asking is that which ones of these would be smaller than a cell, which ones of these would be larger than a cell. And I'm not expecting you to post an answer to the chat to this question. This is kind of the warm up. But what we see, we've talked about the atoms and we've talked about the molecules so far on that course. So clearly these were smaller things than cells. And today, really, we're going to focus for the remaining of the chapter to talk about cell organelles. So these parts that make up the cell itself. So that's going to be our focus. Then later on this course and on your later courses, for example, when you move on to BIO 201, 202, we end up talking a lot more about organs. We end up talking about tissues. On this course, we do get to explore tissues a little bit. But that gives you an idea where we are on the path of looking at life on different levels. The question that I would like to present next is that I have a list of things here, and they are kind of listed in an increasing size. So what I have as A, the subatomic particles, they're the smallest of all of these. And what I have at the very end of the list, I think I have organelles. So an organelle is an organism we could talk about is a thing that exists on its own. So for example, humans are made of 11 organ systems, and that gives us an organism. So what I was wanting to do here was to challenge you to think of what is the turning point where we end up with things that are able to live by themselves. And I'm going to tell you, it's one of those, and above or below that, everything else either is capable of living on its own, or above or below that, everything else still needs others to live by themselves. So what would be the turning point on this list? If you can share that with me on the chat and with your peers, that allows me to take also attendance, because I know we had few people joining a little later, so they also get to do that attendance. So which one of these would be the level of scale after which things are capable of existing on their own? And I hear answers coming in. I'm going to share the screen again. Sorry, I didn't actually mean to not share it. So let me jump back in. But I see that there's a good understanding of what's going on. You should be able to see the screen again. And it really is the cells. Cells and everything bigger than that is capable to live independently. And remember that the earliest life, when we talked about life evolving in the water, those were really simple cells. Those were the prokaryotic cells. And then when we had bunch of cells working together, we were already starting to be able to form tissues, and tissues can form organs, and organs then, when we have a bunch of organs that serve the same purpose, are located kind of working all together for the same reason, that makes our organ system, and we get organelle. So perfect. If you said cell, you're doing great, and thank you for joining me on that. Just a couple of, to get our thinking again going on, we talked about cells being small, and what we saw is that some cells, though, even though most of the cells are small, some cells, though, can end up being large. And by this large, I more of mean like in terms of their length, if we're talking of a 1 meter in length. For example, the neurons, the nerve cells in the spinal cord. The next question asks if it's possible to see all or any of the cell characteristics with a naked eye. And that really depends on where do you draw the line. We can see the nucleus typically. But for this purpose, this question, I would say that we are not always able to see even the nucleus. So I would say that true would be a correct answer. Well, good job for everyone who joined me on this. And I want to jump a little bit backwards to where we were and kind of get into that little stuff inside the cell. And I think we finished last time by talking about the fact that cells, especially the eukaryotic cells, had these specific compartments. Well, now we're going to be focusing for the remainder of the class by looking at those specific compartments. But what's going to become important is that we'll notice that not only the cell itself is enclosed by a membrane, also all these organelles that we find within the cell are enclosed by a membrane. And that membrane has many names. Typically what you end up seeing textbooks using is that they call them these plasma membranes and or cell membranes. But what I like to do with you is to explore a little bit further. Can we understand, based on the name of these membranes, a little bit more what they are and what they do? So what I have here on this yellow color showing up is a section of a cell membrane. So I'm just taking a little piece of the cell's membrane and I expanded it. And what we'll end up noticing is that these cell membranes are made of something known as phospholipids. So phospholipids are these individual components that we have. And then we have a bunch of these phospholipids packed together, and that forms the cell membrane. So let's have a look of phospholipid and understand a little bit more what's so important for us to know about phospholipid and what parts of that can explain us how cell membranes behave. Well, here I have a chemical formula that I've just sketched real fast of what phospholipid is made in terms of all the chemistry that we've learned so far. So let's look through it step by step. So first of all, phospholipid contains a phosphate group, which forms this, if you wish, like head part of this molecule. In addition, there's two fatty acid chains. We often talk about those being the tails of the phospholipid. And in the middle of all that, there is a glycerol. So that gives us a clue where the name phospholipid bilayer comes from and why it might be so good in describing that what plasma membrane really is. So it's made of phospholipids, these very things that we just looked at. And the fact that it's a bilayer refers to the fact that there's two of these layers. So we have two layers of phospholipids that are lining right next to each other. Well, you might wonder, why do we care? Why is there two? Why is there not just one? Well, that all comes back to what we just learned about the composition of these phospholipids. They had and I'm trying to just get a pointer out, they had a head part. And it turns out that this head part, the phosphate group, is very attracted to water. So it likes to be surrounded, or it likes to be close to the water. So it's described as being hydrophilic. So if we take phospholipids and put individual phospholipids in a jar of water, or a jar of any stuff, they always try to arrange so that the heads are facing towards that water. Well, what's interesting is that the tails of the phospholipid actually do not share this love for water. They are actually hydrophobic, so they try to avoid the water as much as they can. So they tend to pack in an arrangement that would help them to get away from the water. Well, one way to do this is really to arrange in a way that we just saw on this diagram, that let's make two layers of these membranes made of phospholipids and have these two layers facing against each other so that these tails are facing against each other. So that way those tails don't have to face any of the water that's located both outside and inside this membrane. And the heads that are attracted to the water are able to deal with this water. So that's kind of a handy thing for us to know in terms of how these membranes are formed and what they do. Well, it's not just the membranes that are phospholipids that form the membranes of the cell. We have a lot of other stuff within these membranes. For example, we have many proteins embedded into these cell membranes, cell walls. And proteins can serve many different functions. They can facilitate movement of materials in and out of the cell so that everything can go through, or only selected things can go through. Sometimes we move things where they normally wouldn't go. So they can be actually transporting material, working a little bit like a pump, moving things where it normally wouldn't go against its concentration gradient. They can be a little bit like a lock that screens that whatever is trying to come in to the cell, that is it allowed to come in? Is it not allowed to come in? They can be recognizing, are we at the right place? Is everything around me still part of that body that I'm part of? They can be serving like enzymes, and we talked about enzymes a little bit last time, and we'll talk plenty more about enzymes later. Or they can facilitate joining different cells, different membranes to each other. So for example, if we have two cells that both have a membrane, these proteins can serve as a way for us to pass messages and also materials from one cell to another by forming this kind of a junction between two cells. So lots of things that proteins can do. So now that we know that cell membranes are made of these phospholipids and proteins and a bunch of other stuff that can be there, we get to the concept that's often discussed as or addressed as describing a cell membrane as a fluid mosaic. Well, this name is really descriptive of what a cell membrane is. And I heard that there was a sound of a question, so I'll jump right to that after I've gone through this part. So if we are looking at these cell membrane walls, they are very much moving constantly, changing. And that refers to the fluid part. So what we see on diagrams, on textbooks, are really just snapshots in time. Instead, really things are constantly moving within that cell wall. They're flipping their locations. Proteins might be moving around, other elements. And also materials are moving across these cell membranes. So rarely is it as stationary as we see on these pictures. Instead, there's a lot moving around. The other thing, the term mosaic refers to the fact that these cell membranes are made of many different components. So if anyone's into arts or anything like that, you might have heard about mosaic pieces of art. These are little pieces of rock or ceramic that we put next to each other, and they all form a picture together. So they're made of many little things that make up a larger whole. So that's where the term fluid mosaic that we use to describe cell membranes often comes from. And that really kind of brings us back to what we saw earlier on, that sometimes we end up in situation where we want to have a larger surface area. So we might want to increase the cell surface area by letting this plasma membrane fold or have microvilli that come out of it. I think that this is a particularly good image. We see a couple of cells actually, and within that cell we can see our nucleus there, but we see that the outermost layer has these folds. So on that note, this is a good time for us to have a look of what sort of questions did we get, and let me just jump to there. And I think that it was just previous messages or messages from other apps. I don't see a new question, but if you do have a new question, this is of course a good time to shout it out or type it. Otherwise, I'm just gonna again soldier on, and we're gonna have a look of the rest of the things that I really want to cover today. So again, you should be seeing my screen. And we're going to have a look now that we've talked about the cell, and that the fact that a cell typically would have this membrane around itself. So we saw that cell is enclosed by a membrane, and actually what we ended up noticing is that it's a double membrane of phospholipids. What's inside this cell then? Well, all of the stuff that's not cell organelles, so we'll go through various different kinds of cell organelles in just a moment, but if it's not a cell organelle, it is cytoplasm. So cytoplasm is kind of a jelly-like stuff that fills the cell. So it really forms this matrix where everything else is kind of floating around. And when I say floating, I don't mean that they're moving too much. That's where the jelly-like fluidity characteristic of the cytoplasm is. A lot of cell organelles, if they are not serving the purpose of moving around, they actually can be fairly well locked where they need to be by a scaffolding that holds stuff in place. And we'll talk about that right at the end. But what cytoplasm does, it really fills rest of the cell. It's also the place where all of these cell activities take place. So we'll notice that cell does a lot of things, and it needs to have a place where it does these things. Well, the place where most of these things take place is within the cytoplasm. That's also where a lot of waste products end up building up, a lot of metabolic processes take place. But overall, it really is just a matrix for everything else to happen. Another thing that cytoplasm does, it does keep the organelles where they need to be, so they don't all end up traveling to, for example, the one part of the cell. So cytoplasm is an important term, an important component of the cells, and that's really all the stuff inside the cell. Well, one of the cell organelles that I assume that you have heard, and that's probably the most likely bet that I would say that all students have heard about, is a nucleus. So nucleus is typically this large rounded blob inside the cell, and that normally stains very dark when we're looking at the cells on a microscope once we have stained them. And I said earlier on that you could just about tell apart a nucleus with a naked eye, but you would need to have good eyes. So let's have a look at what nucleus does and why are we so focused on understanding the important role of nucleus. Well, nucleus is really where the DNA is located at. And in addition to DNA, of course, RNA. And we'll talk about the differences between DNA and RNA in just a second. What you often see on your textbooks are these H or X-shaped structures, such as here. Oh, I didn't mean to run away from that, such as here. And what these are actually chromatin that has condensed to take this kind of a shape that we can tell apart with an electron microscope. It's a mixture of DNA, protein, and RNA. So it's really when the DNA is not just floating around within the nucleus free, but it actually condenses and takes this shape. And there's many elements that are involved in the chromatin to take this shape that we can tell apart. Of course, we remember that DNA was so important for us because that's where all the genes are housed within. So really, it is where instructions for everything in your body are stored on a cellular level at least. So what DNA really does, you can consider it as a massive book of instructions for many, many, many different things. And what these many, many different things are, they are often proteins. So proteins are really the workhorses that make all other things happen within the cell. So if you want to use that analog that I think I've used with you before, that DNA would be like the cooking book, the cookery book, full of all kinds of recipes. Well, we don't want to cook all the foods at the same time. We only want to use the foods that we really need at that time. And what we do, we pick the genes, which are recipes for things that we happen to need at that time. And by those genes, we can prepare proteins that are the exact proteins that our cell needs at that time and at that place. So one thing to note before we start talking about the differences between DNA and RNA is that nucleus is enclosed inside, again, a membrane sac. But what we'll notice is that there are little holes within the membrane that encloses the DNA inside the nucleus. And these little holes become important because they facilitate that stuff is able to leave the nucleus, and also stuff is able to come into the nucleus. So that's something that we want to know and we want to make a note of. So now that we've talked about this nucleus as a structure, I really want to talk about more of the function. What happens? Why do we need to make proteins, and how does that happen? So what we end up noticing or finding out is that DNA is always kept inside the nucleus. It never leaves here past this nuclear envelope, this membrane that encloses the nucleus. So DNA always stays in there. Well, sometimes we might need to take pieces of the DNA and information that the DNA carry out from the nucleus. And if we do that, we make RNA. RNA, unlike DNA, is allowed to leave the nucleus, so it can travel out of there. So that's why RNA becomes important for us. And then this RNA can be used as instructions for making a chain of amino acids. So it really gives us a step-by-step instruction that now take this amino acid and place it next to this line. Then we take the next amino acid and we place it right after that. And by adding these amino acids one after another, we make this long chain of amino acids that then can fold in to be whatever it needs to be, and that is what's going to be our protein. So that's how we make proteins. So there's two terms that we need to talk about when we talk about how DNA gets converted into protein. So the first term that we're going to talk about is going to be transcription. And if you think of, again, let's go back to just thinking of things in a very basic level. What is transcribing? Transcribing is when you take notes of something. A good example would be that if we think that DNA is this amazing cookery book that has recipes for everything you might need, we don't let that cookery book leave from the nucleus. So let's call this nucleus now our library. That's a very special kind of library. You can't check out books. Those are like the special collection within university library. So they are so valuable that we don't want you to take those out. Well, in order for you to get that recipe from that cookery book, you want to make a copy of that recipe. So you transcribe it. And transcription refers to that very process when we convert the information from the DNA into an RNA. So the information that we had, we make a copy of it. And that process, making a copy of the DNA to produce an RNA, and that happening within the nucleus, that is known as transcription. So you're transcribing the information. Well, now we know that this RNA can now leave the nucleus and it travels outside the nucleus and gets together with a structure known as ribosome. What ribosome does, it brings the RNA strand, which contains that, if you wanted to say that it was a transcribed copy of the recipe, well, it actually makes something useful out of that transcribed recipe. It actually makes the food dish. And how it did that was taking these amino acids and based on the instructions on the RNA, piling them one after another until we have a nice protein. So this protein is the end product. If you use that analog, it's the dish. So this process of converting the RNA into actually something useful is translation. So we're really giving a meaning to that recipe. That recipe is going to be useful after the translation is over. So we have something that came out of that. And that's going to be protein. I hope that the difference between those two terms makes sense to you. It's an important concept that we'll take a little bit of a break from, but towards the end of the semester, we come back again. And that's something that I really hope that you feel comfortable at that time. Well, what we're going to do, we're going to go through a bunch of other structures that belong to the cell. But what I want to do is take just a really minute break. First of all, because I need to turn something off. But second of all, it's always good to just take a breather. You can think if you have any questions of anything we've discussed, and we will continue in literally just 2 minutes. Oh, yeah, thank you everyone for sticking around. I know that was a really quick break. Hopefully it gave you a chance to kind of take a break from the material, collect your thoughts, and in case you have any questions. I noticed that my signal is running a little low, but it should pick up in no time. But if at any stage you're having a hard time hearing me, seeing me, hearing the screen, again, just let me know. So we just finished by having looked at the nucleus and talked about the role of that and DNA and RNA. Now it would be a good time for us to go to other cell organelles that we find within the cell. Did we have questions? No. So the next cell organelle that I want to discuss with you is going to be called mitochondria. And you might have heard mitochondria being described as the powerhouse of the cell or the power plant of the cell. And that's for the longest of time what the textbook said, that that's where the ATP, which is the cellular energy currency of the cell, is made. Well, it turns out that there's a little bit more to the story, and it turns out that actually ATP can be made in other parts of the cell, but still at large, mitochondria is the structure where most of the ATP is made. But this is really the part that I love about science, is that we think we know something, but we end up finding that our explanations are still only partial explanations. So there's a lot more to find out. But why mitochondria becomes important is that we do take those carbohydrates that we get from the food. We just looked at those in last week's module. And we are able to turn them into ATP, which is essentially just an energy in a form that the cell can use that energy to drive various different activities. But for mitochondria to do this mammoth task of converting carbs into energy, it's going to need something. And what it needs is oxygen. And it also ends up producing carbon dioxide. So we end up with a need to supply oxygen to the mitochondria. And this oxygen is going to come from outside the cell. And we end up with a need to get rid of carbon dioxide. And that carbon dioxide gets taken out of the cell. So really, respiration, the process of making use of gases and getting rid of gases, happens on this small scale, on a cell scale. It doesn't just have to be at the level of your lungs where oxygen comes in and carbon dioxide leaves from your body. It really is on a cell level where all this happens. And again, we will kind of have a look of this in a little bit of more detail when we talk about mitochondria on action on this course in just a few weeks, I believe. But remember that cellular respiration is really the reason why you're breathing. We need to get that oxygen to those cells and to those mitochondria of the cell. And we need to get rid of that carbon dioxide. Well, let's have a look at the structure of the mitochondria a little bit. You see, it's this little structure, but we can do better than the tiny diagram that I have there. So what we end up seeing is that mitochondria is, again, a structure that's enclosed by cell membrane. And it's going to be a double membrane. In addition to that, within the mitochondria, we have these little folds inside it. So this time we're increasing the cell membrane area of the cell membrane, but rather than building these folds to the outside side, we're building them inside this organelle. And what you see on these two pictures are actually electron microscopy images of mitochondria. And mitochondria, these pictures have been colored so that the mitochondria stands out, but it really does have these folds. So the diagrams that we show to you might look simplistic, but they are based on something. Of course, mitochondria wouldn't have eye and mouth. That's just me trying to make it look cute. So these little folds inside the mitochondria are known as cristae. An interesting thing that kind of relates to something that I used to do for a job, which was to work with forensic side of things, is that we end up finding that mitochondria has its own DNA and its own ribosomes. And actually, mitochondrial DNA is inherited only from the mother, not from the father's side. So we can follow the maternal lineage by looking at the DNA from mitochondria. Well, what's useful about that is that mitochondrial DNA survives usually much better than the other DNA that we might find within the nucleus. So we might be able to get DNA sample where we think that there's no DNA sample to be acquired from. Another important structure that I want to talk about, we end up finding mitochondria both on animal and plant cells, but only plant cells have the next structure that we're going to talk about. And this structure is going to be our chloroplasts. So chloroplasts are sites where photosynthesis takes place. So what chloroplasts do, they take the solar energy that we see here. They take also water, the H2O, and they take carbon dioxide. And they can convert that into glucose, so sugars. What we see here is one of the most simplest sugar molecules, glucose. And as a result of that process, they also make oxygen. So a lot going on there in the process of photosynthesis, and we'll look at photosynthesis in great detail, step by step, as part of this course. At this point, I'm just wanting to show you a general picture. But remember that photosynthesis requires the carbon dioxide, requires the water, and requires the solar energy. And that makes our glucose, which is sugar. And it also makes oxygen as a byproduct. So really, we owe our existence for having chloroplasts in all the plants around us. We utilize the oxygen that they put out. So let's have a look at this minute tiny structure inside a cell able to do this amazing job of photosynthesis. And what I've done here, I've simply added numbers to actually balance out this equation of making equation of photosynthesis. So what we are finding in chloroplast, here's a good picture of what it looks like. Again, it's an artistic impression, so it's not quite accurate, but it's probably better than some of the electron microscope images that we could see. We'll end up noticing that chloroplast is again enclosed by a double membrane. So it's surrounded by two membranes, again making those compartments within the cell. Within this structure, we find this kind of an aqueous fluid that's called stroma. So that fills the chloroplast, this fluid. In addition, we find thylakoids. Thylakoids are these disc-like sacs. We are noticing that when a bunch of these disc-like sacs is piled one on top of each other, that's known as a granum. So those would be some of the terms that are going to be useful when we're looking at parts of the chloroplast. Again, remember, you can use your notes, you can use your textbook when you take your quizzes. Well, the next part within the cell that I wanted to talk about is something that we call endoplasmic reticulum. That's quite a mouthful, so for that reason, we rarely end up writing it out on our notes, and I at least simply describe it as ER. So what endoplasmic reticulum is, it's a network of these membranes or these walls that we have talked. And this network floats or is located within the cytoplasm. So it's connected to the nucleus and the membrane of the nucleus and continues as an outgrowth from there. And really, this picture here is a rather good at showing the variety that we can have within the endoplasmic reticulum. Some of it is smooth, so it means that the surface of that ER just really doesn't have anything attached to it. And this smooth ER performs many functions within the cell. Some of the wall of the endoplasmic reticulum is rough. And by this roughness of the membrane, we mean that ribosomes are attached to that. And what you will remember from what we discussed earlier on, ribosomes were the structures that made proteins. So this is really the core of the process of translation. So lots of the translation happens here at the rough ER. So rough ER has these ribosomes attached to it. Well, the next structure for us to have a look of is known as Golgi apparatus. Golgi apparatus is this stack of membranes and also vesicles, which are these tinier sacs, packed together, folded up together, very much kind of like all arching around each other. And what Golgi apparatus does, I describe it to my students as a distribution center of the cell. So Golgi apparatus receives materials from other parts of the cell, arranges and repackages them, and then sends them out. So really the function of the Golgi apparatus is to take the material, arrange it, pack it, and then send it out back to the cell. So it does that kind of a distribution center, if you think of trucking industry, similar function. Well, what I have next is a list of many different kinds of sacs that we can find inside the cell that serve many other purposes. So we have vesicles, we have lysosomes, and we have vacuoles. All of these either transport material out of some part or material into some part or store some sort of materials, or can be used to store materials that would be so dangerous to have floating around free within the cell. So there are all these little vesicles, lysosomes and vacuoles are little sacs doing a business of transporting, storing, or helping for processes to take place within the cytoplasm of a cell. Few other structures that you might have heard, and if you haven't heard it before, we will talk about these in a great detail later on, but I want to mention them at least. So centrioles are these pair of cylinder tubules that are usually located somewhere very close to where the nucleus is, and they play an important role when the cell is getting ready to divide into two. So when we're pulling those chromosomes apart, so part of the cell is going to get some of that chromosome and other part is going to get some of that chromosome. So centrioles play an important role in that, and we'll talk about them later, but just remember that they exist there. And final structure that I want to mention is called cytoskeleton. So what cytoskeleton does, it's a network of microscopic tiny filaments and tubules within the cytoplasm. And it's going to be everywhere here within the cytoplasm. I just drew it to one part because I ran out of steam to draw it anywhere else. But what they do, they help to provide scaffolding for the cell. So for the cell to hold its shape and allow also materials to move along within that cell. So you can almost think of them as highways of the cell and the cytoplasm of the cell. So these are great transportation pathways for these various different kinds of vesicles to travel and other stuff to travel within the cell. The final difference that I wanted to find out was that there is a big difference between animal cells, which is largely what we have been looking on most of our examples, and plant cells. So the big differences there is how the cell membrane, or the outer layer of the cell is made of. So in animal cells, we have this kind of a plasma membrane double layer, bilayer, that's bending and able to adjust a little bit to the changes in the shape of the cell. Plant cells instead are very rigid, so they remain very much in the same shape, and that's how plants are able to stand up high up. So they have a lot of cellulose in the wall, giving the rigidity and the shape to these cells. Plant cells often physically connect from one cell to another. As we see here, this is a diagram of two cells, and the materials flow from one cell to another through these openings of the plant on the walls of the plant cells. And these openings are known as plasmamodesmatas. Of course, we know that plant cells are going to be the only of these cells that are capable of doing photosynthesis. So they are the only ones that are going to have these chloroplasts present. Mitochondria we find on both. And what I want to finish this session on is by having a look at different kinds of ways how cells can connect to each other. And there's three types that I want to be familiar with. Let's start with tight junctions. So as you see on this diagram, tight junctions are these junctions, these parts where two cells are joining together by a bunch of proteins having packed really close to each other, and they form these continuous lines that are almost like zipper-like lines connecting one cell to another. Desmosomes are sometimes in some textbooks also called as adhesion junctions. Well, they're larger anchoring spots within the plasma membrane where we have many filaments coming out of it. So it really is like a lot of that network within the cell connects to these spots. And there's a physical connection also from one cell to another, from one desmosome to another. Gap junctions actually are proteins that have an opening inside them, through them from one cell to another. And this is where we discussed earlier on that materials could come in and leave the cell. But in this case, now we actually have this connection between two cells, from one cell to another. And that really brings me to the completion of everything that I had planned to share about the cells today. I know it's been a lot to take in, but remember you have those lecture videos available on your weekly learning module. And if you have any questions, I'm going to stick around here to answer to any questions that you might have, clarify any topics. But if you don't, what I would like you to do before you leave, just post your name again to the chat so I can give you a full credit for having stuck around for the entire session. And that's all I have. I'm going to wish you a very good rest of the evening, and I'll see you on Wednesday. We'll look at the lab for this week on Wednesday. Have a good night. Thank you again. Thank you so much. Have a good night. And like I said, I'm around here if you have any questions, but you're free to go. Thank you so much. Have a good night. Yeah, like I said, I'm going to be around. If you have any questions, you can ask them. If you want to ask them outside the recording, I can post the recording. But that's all we have for tonight's session. And for anyone who's sticking around and wondering what I'm doing, I'm just taking the attendance manually. So that's what I'm writing down. But this is the end of this session. So if you have questions, feel free to just unmute yourself, ask on the chat. But otherwise, that's it.